Hello, welcome to the EPG Pathshala. I am Nupur Joshi, Research Scholar in Division of Forensic Science, Galgotias University. Today, I will discuss with you crime scene investigation and management of crime scene, searching methods, documentation and types of physical evidences, importance of evidence. First of all, let us understand the physical evidence. Definition: physical evidence is any tangible material or object that plays some vital role in the matter that gave rise to the litigation introduced in a trial intended to prove a fact in issue based on its demonstrable physical characteristics. Physical evidence in forensic science role. There is a vital role of physical evidence in the investigation of a crime scene. More or less, it all depends on different factors, namely number of physical evidences recovered and how much potential it has. Evidences recovered from the crime spot are very crucial in reconstruction of the events. The physical evidence alone does not describe anything that happened, but it can support or contradict accounts given by witnesses and or suspects. The information obtained from physical evidences recovered can also generate leads to the information and also confirms the reconstruction of a crime to a jury. Now, how to process the physical evidences? There are myriads of steps involved and to be followed while processing the physical evidences. These are stated under: first, discovering physical evidence; next, recognizing the physical evidence; third, examining the physical evidence; fourth, collecting the physical evidence; fifth, recording the physical evidence; sixth, identifying the physical evidence; packaging, conveying and storing the physical evidence; exhibiting the physical evidence in court; disposing of the physical evidence when the case is closed. Now, types of physical evidences. Different types of physical evidences recovered at the crime scene. A few of them have been discussed in depth below. The first is blood, second is semen, next is saliva, documents, next explosives, firearms and ammunition, fingerprints, DNA evidences, skeletal evidences and petroleum products, and plastic bags, plastic, rubber and other polymers, vehicle lights, wood and other vegetative matter. Now, let us throw some light on a few crucial physical evidences. Blood is a bodily fluid in animals and humans that delivers essential substances, namely nutrients and oxygen, to the cells and transports metabolic waste products away from those cells. Analysts investigate and study the patterns at the scene and often screen and profile the blood in the laboratory as well. It has become more common for blood stain pattern analysts to have a degree in math or a physical science such as biology, chemistry or physics. Blood is often encountered at many crime scenes, specifically in heinous crimes, for example, murder, rapes, suicide, hit and run and many more. Many potential details can be discovered from blood evidence regarding the crime scene, which can further be used in the DNA in the investigation. DNA proteins and cells are the vital components of the blood, allowing the laboratory to perform the examinations. Plenitude of useful information can be ascertained from blood. The examinations can be performed to ascertain the source of origin, if the source of origin of blood is human or non-human. Scientific Animal family. The specific animal family can be determined for non-human blood. Exclusion and inclusion of possible suspects. If blood belongs to human origin, then it can be compared with the specimen blood for exclusion or inclusion of the possible suspects. Next physical evidence is semen. Semen or seminal fluid is an organic fluid that may contain spermatozoa. It is secreted from the sexual glands of male or hermaphroditic animals and can fertilize female ova. In case of sexual offences and when the perpetrator is a male, then seminal stains are common to encounter from the crime scenes. The stains may be found on the body of the victim, clothings, rags, beddings and multitudes of sources as well. So, let us focus on few steps involved in the collection of semen stains from the crime scene. So, now let us focus on some few steps involved in the collection of semen stains from the crime scene. All the stained material under suspicion should be recovered carefully in order to avoid cross-contamination of evidences and to prevent their loss. Each item should be separately packed. All damp stains should be air dried. The location of moist stain on the evidences itself must be marked. On air dry, it may not be visible. Use clean paper, spread it under the item to catch any kind of debris which may be dislodged during the drying process and between items hanging next to each other to prevent cross-contamination. Avail assistance of forensic staff to recover the stains in case when semen stains are on objects that cannot be easily submitted to the laboratory. Next physical evidence is saliva. Now, what is saliva? Saliva is a complex glandular secretion made by the salivary glands found in the underlying tissues present in the oral cavity. The basic unit of salivary glands are clusters of cells called acini which secrete a fluid containing water, electrolytes, mucus, enzymes, etc. The fluid flows out of the acini into collecting ducts. Within the ducts, the composition of the secretion is altered, such as much of the sodium being actively reabsorbed, potassium is secreted and large quantities of bicarbonate ion are secreted. The fluid goes through small collecting ducts within the salivary glands which lead into larger ducts, eventually forming a single large duct that empties into the oral cavity as saliva. Assists in lubrication and serves to protect the oral surfaces, providing moisture and lubricant to materials in the mouth, for example food, dissolving chemicals that can stimulate the taste buds and provide sensory information about the material, initiating the digestion of complex carbohydrates before the material is swallowed, for example salivary amylase. Bicarbonate and potassium secretion is of tremendous importance as it provides a critical buffer for neutralizing the massive quantities of acid produced in the four stomachs. A few points must be kept in consideration while collecting the saliva evidence. If the saliva stained object is transportable, then it is advisable to submit the object intact, and in case it is not possible to transport the stained object, example bite mark, on a body, then following points might be kept in mind while collecting the saliva stain. Moisten a sterile cotton swab with distilled or tap water. Shake the swab to remove excess water. Gently swab the suspected saliva stain. Allow the swab to thoroughly air dry prior to packaging in a paper envelope and seal. In order to collect the negative control, select an unstained area and collect the sample in the same manner as described before. This swab will serve as a negative control. Next physical evidence is documents. The section 3 of the Indian Evidence Act describes document as Document means any matter expressed or described upon any substance by means of letters, figures or marks, or by more than one of these means intended to be used or which may be used for the purpose of recording that matter. A document examiner is often asked to determine whether a questioned item is originated from the same source as that of known items, followed by presenting their opinion on the matter in court as an expert witness. A document examiner also determines what has happened to a document, its absolute relative date of production, decipher information on the document that has been obscured, obliterated or erased. Now, types of documents that are examined. Documents plays a vital role in our daily lives, including business and personal affairs. The authenticity of almost any kind of document can be questioned. In the broadest sense, a document is anything bearing marks, signs or symbols which conveys message or meaning to someone. It includes traditional paper documents like graffiti on a wall, stamp impressions or covert markings hidden in a written letter, among other things, etc. A questioned document may be a sheet of paper bearing handwriting or mechanically produced text such as a ransom note, a forged check or a business contract. That is to say, it may be some material which is not normally thought of as a document. A forensic document examiner conducts examinations and comparisons which can be quite diverse. The following examinations are usually performed by a forensic document examiner. The first, handwriting and signature typewriters, photocopies, printers, inkjet printers, fax machines, check writers, rubber stamps, price markers, label markers, printing processes, ink, pencil, paper, alterations, additions, erasures, obliterations, indentations, detection or decipherment, sequence determination, physical matching. Handwriting examinations. Generally, there are 3 stages in the process of handwriting examination. In brief, they are analysis, comparison, questioned item against the known standard and evaluation. Optionally, the 4th step consisting of verification or validation or peer review may also be there. Now, tools for document examination. Excellent eyesight, hand lens, stereo microscope, electrostatic detection device, that is EDD, video spectral comparator, that is VSC, DocuBox Dragon, DocuCenter, Raman spectrophotometer, etc. Explosives. An explosive material, also called an explosive, is a reactive substance that contains humongous of potential energy that can result into an explosion if released suddenly, usually accompanied by the production of light, heat, sound and pressure. That is to say that an explosive is a substance, may be an element or a compound or a mixture which is capable of exerting pressure on its surroundings on explosion or transformation. Explosives are studied by forensic personnel mainly related to mass destruction, episodes where bombs are used for illicit activities. The explosive residues discovered from the crime scene are examined for many causes, especially so as to identify the explosive material, the source and intention of the explosion. An explosive has myriads of applications which are legitimate and do not cause any harm to any animal or any human being. Now, the legitimate uses. Legitimate uses of explosives. An explosive might be used in blasting rocks for mining, oil explorations, in satellite and spacecraft propulsions, in constructing roads, railway lines, etc. in fireworks display and may also be used as military explosives, which we will discuss later. Now, illegitimate uses are: the criminals are using the explosives for causing havoc to individuals or a nation by blasting bombs. The illegitimate use of explosives causes high destruction to the integrity of any nation and is severely punitive under the Indian Penal Code, Explosive Act and the Explosive Substance Act as well. Some common examples of explosives are RDX, TNT, TETN, ANFO, dynamite, etc. Forensics plays a very important role in the investigation of explosions where explosive substances or materials are the prime ingredients. Explosives can be detected prior to explosions and also in the wake of the explosion by forensic spot tests and also by high-tech forensic analytical tools. In the simplest term, we can define an explosion as rapid increase in volume and release of energy along with the generation of high temperature and release of gases. Due to the presence of organic compounds containing NO2 and NHNO2 groups and others, an explosion is a spontaneous chemical reaction which is driven by the release of heat and energy. This type of explosion is called the chemical explosion. The chemical explosion is of three types, namely decomposition, deflagration and denotation. The chemical decomposition of an explosive is a slower process which takes place in storage. This may take years, days, hours or a fraction of a second. Deflagration and detonation are two rapid forms of chemical decomposition. Deflagration of the explosive material is propagated by a flame front which moves slowly through the explosive material. Low explosive undergoes the process of deflagration. Detonation of an explosive is propagated by an explosive shock wave traversing the explosive material. The shock front is capable of passing through the high explosive material at great speeds, typically thousands of meters per second. Detonation takes place in high explosives. The explosives can be classified on the basis of composition, velocity, sensitivity and physical forms. But broadly, explosives are of three types: low explosives, high explosives and miscellaneous, which can be further divided into home-made explosives, nuclear explosives and others. Availability and cost, sensitivity and sensitivity. 2. Initiation, velocity of detonation, stability, power, performance and strength, brisance, density, volatility, toxicity, explosive train, oxygen balance and chemical composition are some of the important characteristics of an explosives which are very important to ascertain whether the explosive is suitable for a particular use or not. There are some important rules which an investigating officer must keep in mind for evidence collection. A few of them are mentioned below. Make sure that there are no suspected devices. Use aerial photography. Use wire mesh screens to collect post-blast residues. Do not ever handle the potential explosives yourself. Clear and secure the area from unauthorized persons. Call the bomb squad. Use the bomb disposal suit. Always photograph the item as found. Always note the evidence and its location. Do not restrict to a limited area of blast scene. Always wear latex gloves to collect the evidence from the crime scene. Search far and wide from epicenter. Always use clean, suitable and unused containers. Always label properly. Always change gloves between collecting different items. Always clean the tools between collecting different items. Always keep the evidence in a secure location on the scene. Always maintain the chain of custody. The size of explosive does not matter. Collect soot deposits interview the witness, search for explosive device or bombs, fragments. Ballistic evidences such as firearms and ammunition. The range of evidence in firearms related cases can be as small as a piece of bullet fragment having rifling marks, or as large as hundreds of bullets and cartridge cases and numerous firearms. A firearm is a portable gun, being a barreled weapon at the launches one or more projectiles often driven by the action of an explosive force, whereas ammunition may be defined as a propellant and projectile or anything that can be used in combat including bombs, missiles, landmines and anti-personnel mines. A ballistic expert is a forensic specialist who is responsible for accumulating and analyzing the ballistic related evidence, which includes firearms and ammunition. The ballistic evidence may encounter at the crime scene may include firearms, spent cartridges, spent shell cartridge or bullets, shot shell wading, live ammunition or clothing. Firearm evidence collection. Firearm evidence can be recovered in plenitude of ways and areas. Firearms themselves can be recovered at shooting scenes by crime scene investigators and sent to the laboratory. Bullets, bullet fragments, cartridge cases, shot shell wadings, etc. are normally collected individually after proper documentation, photography and sent to the laboratory. Bullet evidence can also be obtained at autopsy or in an emergency room setting. In these cases, the sample should be marked as a biohazard and then sent to the laboratory. Each laboratory has written procedures for packaging and submitting evidences. Bullets or slugs that do not strike a person are often embedded into a nearby surface such as wood or drywall. This evidence is the best gathered by cutting out a section of the material and submitting it to the laboratory to allow a firearm examiner to carefully extract it. This prevents adding or destroying any markings that could be crucial to identifying or matching the suspected firearm. Next, analysis of firearm evidences. A well-trained firearms examiner should perform the evaluation and comparison of this evidence. The marks left on ammunition may help in determining which firearm was used to fire the bullet. From the bullets fired to calibers and rifling patterns, it is possible to identify the characteristics of the firearms. Cartridges and cases are analyzed to search for signs of firing, pin impressions, ejector marks, extractor marks and other tool marks. Even from small samples, information can be developed to indicate the type of firearm used and possibly identify the actual firearm that was used. Other firearms evidence that could be found at a shooting scene includes shot shell, wads and shot pellets. These can indicate the gauge of the shotgun. Wads and pellets can be gathered and preserved in the same manner as bullets and cartridge cases. By examining the wadding materials, the examiner may be able to determine the gauge of the shotgun, the manufacturer or the marketer, a range of possible shot sizes based on impressions in the shot shell wad, individual characteristics in some cases. So, now we start with the crime scene investigation review key. First, isolate and secure the scene, document the scene, search for evidence, collect and package evidences, maintaining the chain of custody. submit evidences for the analysis, medical examiner, initial walk-through plan of action, date, mention date, time, location, scale, recovered items, fixed point distances, compass, legends, from to fixed point, wall, door, window, grid, quadrant or zone, spiral line or strip. By examining varying materials, the examiner may be able to determine the gauge of the shotgun, the manufacturer or marketer, a range of possible shot sizes based on impressions in the shot shell wad, individual characteristics in some cases. So, I would like to conclude that it is very important to preserve the crime scene at that same point of time, and if the crime scene is evaluated and looked for in a very systematic manner, can give very good results. That is all for this section. Thank you.